So today we'll talk about transcription in a lot more detail, and then for Tuesday we'll leave translation. Before we do, this is where we left it. We said that the structure of a generic gene is a little more than the region that is transcribed into RNA. We need to include the promoter sequences that will tell RNA polymerase where to begin transcription and in which direction to go. There is some sort of termination of transcription information in the form of a termination sequence in prokaryotes and in some eukaryotic genes, or in the form of a poly-A signal that gets transcribed into that primary transcript and later used to process that primary transcript to form a poly-A tail. And today we'll barely touch on some sequence that is within the coding region that gets transcribed into that RNA transcript, and that in eukaryotes is used by cells to instruct on how the splicing out of introns should take place, okay? So a little more than the sequence that you will see in that mature RNA molecule, whatever that molecule might be. So here's a brief overview of the stages of protein expression, even though we made such emphasis on non-coding or non-protein coding genes on Tuesday. Now we're going back into this mode of thinking that genes code for proteins. Again, not always, but we'll discuss about genes that do code for proteins because we need to get through both their transcription and their translation. So these are the stages of protein expression, and whenever you see something in the middle, in between bacteria and eukaryotes, all that means is that they both do it. And whenever you see something that is on one side or the other, that means that it's primarily one type of organism or the other. So both have to recognize promoters, initiate transcription, and elongate those RNA transcripts or chains. Bacteria will actively terminate the transcription of those RNA molecules, again, of protein-coding mRNAs, whereas eukaryotes don't. They use this polyadenylation mechanism that we'll discuss later. And after chain termination, bacteria jumps straight into translation, whereas eukaryotes need to do further processing of those primary transcripts. So we'll go through all of these stages in order. We'll start with promoter recognition. And we'll start the simplest that there is, and that is promoter recognition in prokaryotes, in bacteria. So what happens is, we already spoke about these bent arrows here used to indicate TSS or +1 side of transcription start. What happens is 10 nucleotides before that TSS, there is what we call the -10 region. So it's 10 nucleotides upstream of that TSS. And there are these sequences that are signature sequences or consensus sequences. When we say consensus, it can be a little misleading. So who's the consensus among, right? So is this consensus among scientists? Is it a consensus? What we mean by consensus is that many, if not all, bacterial genes will share these sequences. So if anything, there is a consensus among bacterial genes to have these TAT, AAT sequences, 10 bases upstream of the TSS. And then there is another one, 35 bases upstream of the TSS that we call the -35 region, okay? Now these two, -10 and -35, normally we refer to them collectively as the -10, -35, as a combo of sorts. What they do is they mediate the binding of a very special protein called the sigma factor that can read these sequences when they're present and when they have that very exquisite spacing between them. So not only do these sequences have to be present upstream of a TSS, but they also have to be spaced exactly 25 nucleotides between them. So when that happens, this protein will biochemically interact with these sequences, and once it is bound to DNA, it can go ahead and recruit the RNA polymerase enzyme. And together, the sigma factor and the RNA polymerase will form what we call the holo enzyme, meaning the enzyme as a whole.
Is the -35 and -10 region, is it always that code? Like, so for -10, is it always T?
Yes, yes. These are sequences that have been conserved throughout the evolution of bacteria. And while we're at it, why do you think they have to be so precise? Why can't they differ in sequence? I honestly am not a bacterial geneticist, so I don't know if they can never, ever change a little bit. But for now, let's say that those are pretty strict consensus sequences. So why do you think that they have to be so strict? What could be constraining changes in these key sequences? Yes?
It has to be something that doesn't show up in other parts of the genome, because otherwise it would get confusing for the sigma factor.
Okay, so let's go with that. So if these sequences happen just anywhere, it would confuse the sigma factor, right? We need to understand, though, that they would have to happen together and with the right distance between them. So the probability that this thing would just happen just because, randomly, are a lot lower, right? But the question here is slightly different. Let's say that we have these two and that we change that G there. We change this blue pointer, sure. That G to a C, right? That would, quote unquote, kill that -35 side, right? But why would it kill it? Why wouldn't the sigma factor be able to see that -35 anymore? What does the sigma factor have that it can read these sequences and say, oh no, there's no G there, there's a C now, so not for me? Does the sigma factor have little eyes that can read letter words? Nah. So how does it do it? We kind of spoke about it. Polarity of it. This is your time to shine. Say amino acid polarity. Anyone? How can proteins read sequences in DNA? This is key. And we'll revisit this when we talk about expression regulation or gene expression regulation. But the core concept in all of this is that proteins interact with DNA in a sequence-specific manner, meaning they won't interact with just any region of DNA, but with very specific sequences because of their biochemistry. The biochemistry of the proteins that interact with DNA in a sequence-specific manner is such that they will find those regions with very specific sequences because there's a certain profile of amino acids in their structure that matches nicely with the biochemical profile of these words. So for us, T, T, G, A, C, G is just a bunch of letters. But for the sigma factor, that represents a specific 3D landscape of charges and polarities and atomic masses that they can, like a key and a log with substrates and enzymes. You guys have probably heard these before. Heard this before in the context of enzymes and enzymatic reactions. So just like a key goes perfectly into a log and opens it, same here. There has to be that matching between the biochemical profile of a protein interacting with DNA and the sequence that it interacts with. So these sequences have remained the same for millions of years and still remain the same because they're constrained by the biochemistry of the sigma factor. Because the sigma factor doesn't change, these sequences won't change. If the sigma factor ever changed, because not all sequences can change accordingly immediately overnight, then chances are that one particular bacterium will die and be selected against. So there is this sort of evolutionary constraint between these sequences and the sigma factor, and they continue going together throughout evolution. Does that make sense? All right, so we have our sigma factor bound to the -10, -35. They recruited RNA polymerase, they formed the holoenzyme, and now that everybody is there at the TSS, they can initiate transcription. My question to you is, what would happen? So I mentioned that there is this minus 10, minus 35. What would happen if there is a mutation that doesn't affect the words at those positions, but adds, let's say, 4 letters to just upstream of the TSS, right? So what would happen with the expression of that gene? What would happen to the expression of a gene if a random mutation inserts 4 extra nucleotides between those positions? Because he has to walk 4 more nucleotides or something like that, or because the binding is not as strong, or
Because the binding is not as near, I guess, to
near the TSS. So now this holoenzyme is bound there, but it's having a harder time reaching that TSS and starting transcription. So it will happen, but at a lower rate. Okay, I can work with that. So that's a possibility. What I asked, and no one seemed to agree with that idea, is that the expression of this gene is gone. That this gene is, from that point onward, completely non-functional. So no one seems to agree with that. What you're proposing is the next best thing, right? So yeah, expression of the gene is there, but it's at a lower rate. Any other ideas? Yeah.
Would it be that it's still binded to the promoter,
Uh-huh.
or it would still go on, but now it's 4 digits off, it's 4 nucleotides off, so now everything is off?
Okay. If I may paraphrase, so what you're saying is it's still happening, but it's just shifting 4 nucleotides? Does anyone agree with that? I see some heads, maybe, some heads nodding. Yeah.
That's what I thought would happen too, but now I'm second guessing myself because of the transcription
Star sign.
Yeah.
All right. Drum roll. That's what happens. Transcription will be just the same, but it will start 4 positions earlier. And here's why. That's my little pun there. We shouldn't put the cart in front of the horse. It's not the TSS what determines where the minus 10 and the minus 35 should be. Is wherever the minus 10, minus 35 combo is, what dictates where the plus 1 will be. In other words, if sigma sees a minus 10, minus 35 at the right distances between them, it will bring RNA polymerase, and RNA polymerase will start 10 nucleotides after that minus 10, wherever that happens to be. So if we added a few nucleotides between what used to be the minus 10, minus 35, that now presumably became the minus 14, minus 39, no, they will still be minus 10, minus 35. What changed was the position of the plus 1. Because it is not the TSS that tells sigma where to bind, it is sigma that tells RNA polymerase where the TSS should be. Did that make sense? Yeah?
So technically, we're right in saying that it could be like non-functional because if it's stop codon appears.
But the thing is, stop codons and all of that are going to happen much downstream. Everything, this is something that may sink in a little better on Tuesday, but everything that happens between the TSS and the actual start codon of translation is what we would call the 5' untranslated region. It's a whole bunch of nucleotides that are never translated into protein anyway. And those 5' UTRs are a lot more flexible to changes in size, nucleotide composition, sequence composition, because they're not coding for almost anything. There is information in them, and we'll study some of that, but mostly in a couple of weeks when we talk about gene expression regulation. But for the most part, you're not breaking any code, because the protein coding code starts with the famous AUG that I heard some of you mention before. This is not the AUG. That is not the AUG. That is just the TSS is where transcription starts. The famous AUG is where translation begins. That's going to be downstream. Okay? So everything gets shifted by 4 positions. There's a new TSS that moved over a few nucleotides, and there's an exceedingly high chance that the levels of expression, the timing of expression, the conditions for expression of this gene, none of that changed. The only thing is that there's going to be those 4 extra nucleotides in the 5' UTR. Okay? All right. So what does transcription initiation look like in eukaryotes? It's not much different, except that there's a lot more diversity. So for a while, the thinking was that just like there was the -10, -35 in bacteria, there would be something similar in eukaryotes. And it turned out that people started finding that TAA sequence in promoters of eukaryotic genes. So they thought, okay, excellent. This is like our -10, right? There was no obvious -35, but there was that TAA, and they called it the Tata box. The Tata box, okay? And what research showed is that just like there was a sigma factor binding to the -10, -35 bacteria, there was something called the TBP, that stands for Tata box binding protein in eukaryotes that would bind the Tata box, recruit not RNA polymerase directly, but a whole series of transcription factors from the TF2 family. So first it would be TF2D, then TF2D would bring TF2A and B, and then a whole bunch of other family members. And eventually, when enough of them had piled up at that promoter, they would recruit RNA polymerase, and our RNA polymerase, we wouldn't call these a holoenzyme in prokaryotes because the composition of these complex is more varied in eukaryotes. It's not always the same composition, but we do call these the transcription initiation complex. Right? So there would be this initiation complex that would let RNA polymerase begin transcription. For a while, we all thought that it was going to be as simple. Then it turned out that more research showed that some genes did not have a Tata box, and yet they could be transcribed. And with more and more research, what people found is that there are different kinds of consensus sequences. There is the Tata box, there is also the CAT box, there's GC-rich boxes, and a handful others. And the different combinations of these things can form a functional promoter or core promoter. So here are real examples of the composition of consensus sequences for the promoters of these particular genes. Details obviously don't matter, but what you need to appreciate is how diverse these core promoters are. Some of them have nothing but GC-rich boxes. Some of them have a Tata box, but some don't, and so forth. And the idea is that, again, these consensus sequences will bring in specific members of this TF2 family of proteins, and once enough of them have piled up, they bring in RNA polymerase, forming the TIC, or transcription initiation complex. One other thing to note about eukaryotes is that eukaryotes will use RNA polymerase II to transcribe protein coding genes or transcribe mRNAs. The fact that we call it RNA polymerase II is kind of telling you already that it's more than one, so there's 3, right? And this table here lists the types of RNAs that each of these RNA polymerases use. So, to recap, conceptually, both prokaryotes and eukaryotes use the same approach, and that is identifying key consensus sequences in the core promoters of genes through the binding of specific proteins to them that will either directly in bacteria or more indirectly in eukaryotes bring in an RNA polymerase. In the case of bacteria, there's only one, so that's the one. In the case of eukaryotes, it's RNA polymerase II. And once everybody is where they should be, transcription can begin. Any questions about any of that? pretty straightforward, except that we did touch on something that is kind of important, this idea that genes, and we'll revisit this in a way when we talk about the translation code. There is no, when we say TSS is here, when we say a specific consensus sequence from the core promoter is here, none of those things have necessarily markers to tell the rest of the cell, hey, by the way, here's a core promoter. These things happen the other way around. So there is all these sequences that when in the right combination, they will recruit the various proteins that will make a functional complex. So if I may go back to something that you were suggesting before, that I can't remember your exact words, but you said something like, if there was one of these sequences, the -10, let's say, somewhere else in the genome, and it's all by itself, that would confuse the sigma factor. Yeah, that in a way would happen, except that it's not that the sigma factor would get confused necessarily, it simply wouldn't bind there, right? But there was nothing that was telling the sigma factor, hey, by the way, here's the -10, but you're missing the -35, so don't even bother. No, what's going to happen is the sigma factor will likely touch on that region of DNA, recognize the -10, immediately tell that there was no -35, and move. Just move away, move along, go to a different location, right? So sometimes we speak about these things as if there were some sort of marker in the DNA sequence or in the RNA sequence later on to tell cellular machinery, here's what you should do, here's none of that. So it's all from within these sequences that things start happening, and when you have enough of these proteins coalescing together, for instance, that's when functionally the cell will know that that's a core promoter of a gene that can be transcribed. Okay? All right. So that was just the core promoter. I threw the word core promoter a couple times. I know that we mentioned it before, but just to reiterate, the core promoter is where the transcriptional machinery assembles, right? But genes, both in bacteria and eukaryotes, hence the diagram is shared between the two. In addition to the core promoter, they will have what we normally call regulatory promoters. These are collections of sequences that are also bound by very specific DNA binding proteins or sequence-specific DNA binding proteins that will either facilitate or somehow inhibit the assembly of the transcriptional machinery, of the holoenzyme in the case of prokaryotes or the TIC, transcription initiation complex, in eukaryotes. Right? And we call them regulatory promoters because these are the sequences that, through interactions with these key proteins inside of the cells, they have the ability to regulate the expression of a gene. So the core promoter takes care of telling the cell where the gene should be transcribed and in what direction, but the regulatory promoters are the ones that say, you should transcribe now, but not yet. Maybe you should transcribe some more of this. Maybe you should transcribe, but not as much. Those that are kind of regulating the levels and the timing of transcription. So there is a short video in Canvas about regulatory promoters and enhancers, and I recommend that you check it out. We'll revisit this when we talk about gene expression regulation, but there's a sneak peek there. All right, so our core promoter has assembled the either holoenzyme or the TIC in eukaryotes, and transcription has initiated. What comes right after is chain elongation. And this is sort of the very mechanical, very procedural series of steps that RNA polymerase goes through to make an RNA molecule. And it's very similar in both bacteria and eukaryotes in that there is an unwinding of the double helix, of the DNA double helix. In RNA polymerase, starts reading the template strand and writing an RNA molecule that is anti-complementary to that template strand, and that's that. That's how RNA polymerase initiates transcription. Unlike what you may remember from when you studied DNA replication at some point, RNA polymerase does not need a primer. It doesn't need a little bit of sequence that had been pre-assembled for it to take it from there. No, RNA polymerase can start from scratch. Okay? And in both bacteria and eukaryotes, there's some sort of, it varies from gene to gene, but there is a handful of nucleotides that are added at first very slowly. And then at some point, and when enough of a mini transcript has been synthesized, that's when RNA polymerase lets go of all of these things that were binding to it, the sigma factor in bacteria or the TF2 family proteins in eukaryotes, and takes off and it starts transcribing much faster. Right? So it's like there is a slow start and then it explodes. Okay? All right. So how does elongation go? So you may have seen this before, but again, if in this cartoon we have the DNA template strand on top, right, and we have a series of nucleotides there. Notice that the DNA template strand is written 3' to 5' because that's the direction the RNA polymerase will normally read it. And then RNA polymerase is synthesizing the RNA transcript down here from the 5' end to the 3' end. And the way that works is there's going to be an incoming nucleotide, ribonucleotide, that comes in, sees the base that is exposed on the template strand, tries to base pair with it, respecting base complementarity, the Chargaff's rules, and also the anti-parallel orientation. And if there is successful base pairing, that nucleotide can hang out there long enough that the RNA polymerase says, okay, this nucleotide belongs here, so I'm going to stitch it to the very end of my growing RNA molecule. Right? So it's adding one nucleotide at a time, as long as that new nucleotide can hang around for long enough. Okay? Now, one thing that you may remember from studying nucleic acids before, the nucleotides in both a DNA strand or an RNA strand are linked together by phosphodiester bonds. These are phosphate groups that link nucleotides together, right? But the incoming nucleotide is a triphospho or a triphosphate nucleotide, right? And that matters because it turns out that the energy that is stored chemically in these triphosphate groups is the energy that is used to catalyze the reaction of stitching the new nucleotide to the end of the growing RNA chain. So that's an enzymatic reaction that will require some energy. And I don't know if you guys remember from previous coursework that often when there are enzymatic reactions in cells that require some type of energy, cells will use, do you remember what molecule they use? ATP, right? So ATP is that energy currency of the cell. There's that metaphor thrown around a lot in books and whatnot. ATP is such an energetic molecule that when broken down, when metabolized, it can offer all of this energy that then certain enzymatic processes will use. Well, it turns out that anytime you see an A in RNA, that A came from an adenosyl ribosyl triphosphate, an ATP molecule. So the very same ATP that fuels up muscles and motor proteins and all kinds of endergonic processes in cells is the same ATP that will be broken down and added as an A in an RNA molecule. Same molecule. Some of you may have known that. Some of you may have not, because normally these things are taught separately. GTP as well. So we talk a lot about ATP because ATP is used in all of these processes, and it's true. But from an energetic point of view, they're all equivalently energetic. So ATP, GTP, UTP, and CTP are all super high energy molecules. And in all cases, the RNA polymerase will break that phosphate group, obtain all of that energy that was stored in that phosphate bond between the alpha phosphate and the beta phosphate, and use that energy to catalyze the covalent bonding to the end of that growing RNA. Okay. I think, I forgot that I had this as a question for you guys. But bottom line is the energy for RNA synthesis is stored in the very building blocks that the RNA polymerase molecule uses to make that RNA. Okay? So there's no need to bring any extra energy from the outside. Oh, and it turns out that, okay. Do you know this guy? That's just a cartoon of what I was saying before. So that is the triphosphate form of A in RNA, and that is ATP. Okay? Same molecule. Again, I don't know about you, but when I learned this stuff many years ago, it was ATP was all over the cell when it came to respiration and photosynthesis. And so everything was ATP, ATP, ATP. And then when people spoke about DNA or RNA, no one made the connection from it. Right? It seems like it was two completely different universes, and yet they share that molecule. Questions, comments, clarification? Yeah. This guy, meaning this molecule. Yeah. And what I was saying is that this molecule, as you see it here, you see it, and you recognize that it's one of these building blocks that will make RNA, right? And in particular, this one will add an A to the RNA molecule, right? So this one is the one that adds A to the molecule. And it turns out that this molecule is ATP. And the reason I was bringing that up is because we're all very comfortable, very familiar with the idea that ATP is a super energetic molecule. Rarely do we make the connection that it is that same ATP that gets used to make RNA. And that GTP, UTP, and CTP are equally energetic because they store all of that chemical energy in those triphosphates. Okay? And that's the famous phosphorus that Hershey and Chase made use of when they incubated their bacteriophage with radioactive phosphorus, because there's phosphate in nucleic acids, both RNA and DNA, but not in proteins.
And the other one's adding a U.
Exactly. So this one would be, in fact, that's the one that you see in the cartoon. So this one would get added as a U. And in this case, it would get added as a U to the RNA that is growing because on the template strand, you find an A. So the U will base pair nicely with the A, and it will still have its 3 phosphates attached to it. When RNA polymerase detects that there is nice base pairing with the nucleotide in the template strand, it says, okay, I can add this guy to my growing RNA, and it will catalyze that reaction such that it uses the energy stored in this triphosphate to create a new bond between the last RNA nucleotide in this RNA that is growing to the new nucleotide that just came in. Yep. Okay. Let's move along. So this process continues until in bacteria there is a signal that tells RNA polymerase you should stop here. And that's what we call chain termination, and we'll discuss that now. So there's 2 mechanisms for transcription termination in bacteria. One is called Rho-dependent termination. And the way it works is polymerase is transcribing. At some point towards the end of that gene, there's going to be a sequence that is written in DNA called the Rho utilization site. So raw utilization, the U and T in utilization, they all get combined into RUT. So that RUT sequence gets transcribed into RNA. And now that it's there in RNA, there is this raw protein that can see the RUT sequence in RNA. It couldn't see it in DNA, but it can see it in RNA. And it binds to it. And once it binds to it, it does something funny. It starts moving downstream that RNA molecule. It moves along it, just like RNA polymerase moves along the template strand, this thing moves along the RNA transcript. And it goes much faster than what RNA polymerase goes on the template strand. So it catches up to it. It catches up to it and it physically bumps it off the template strand. It's like almost like bumping cars. So when RAW sees that RUT sequence in RNA, will bind to it and start moving downstream fast until it bumps into RNA polymerase 2, bumps it off DNA. And that's it. Now that RNA polymerase is off, it cannot come back. It doesn't have a sigma factor telling you where to bind or anything. It will go find the next core promoter where it will start doing its job again. But when it comes to this gene, that's it. Termination of that gene is terminated. Sorry, transcription of that gene is terminated. Okay? That's one mechanism. The other, by comparison, is called raw independent termination. This one is a little trickier to understand conceptually. But the idea goes as follows. It turns out that near the end of this particular bacterial gene, there is 3 key sequences that are all written in DNA. Okay? One of them is a stretch of nucleotides that are rich in C's and G's. Okay? Then comes a little spacer, and then comes the same sequence, but in reverse complementary orientation. So in the example here, I'm going to zoom in so that you can see it. In the example here, we have TCG, CCCG, a little spacer sequence that could be anything. And then you have the complementary sequence in reverse orientation. What I mean by that is, if we start reading backwards, we see A, that would pair with T, G, that would pair with C, C, that would pair with G, the 3 Gs, that would pair with the 3 Cs, and the C, that would pair with the G. So there is complementarity and also reverse orientation. What happens is, when these very unique sequences get transcribed into the RNA molecule, now when that RNA molecule that's moving in space happens to bump onto itself, these sequences that are part of the same RNA transcript and that are reverse complementary would love to form one of these intramolecular stretches of double-stranded RNA that we mentioned the other day, right? Think of it as a zipper. So the second that these two molecules kind of see one another, it's almost like a zipper quickly going up. And what happens when you close the zipper? The two sides of your jacket, let's say, come together, right? Same here. You go like this, zip, and these two molecules will want to come together. Now, I emphasized before that these reverse complementary sequences with a little space in between are CG rich. They don't have to be a specific sequence of C's and G's, but just have a lot of them, way more than T's and A's. Why is that? Because throughout evolution, sequences that are CG rich and that have reverse complementarity are much more likely to violently form a double-stranded RNA stretch. Why? Because like in DNA, and I don't know if you guys remember this, but like in DNA, C and G form 3 hydrogen bonds, whereas DNA form only 2. Right? So the very nature of these base pairs makes it such that these zipping up of the, we would normally call these a stem loop or a hairpin because it looks like one of those really old-fashioned hairpins. But the formation of these stem loop structures will be very violent. And And I keep saying that, and it sounds like a joke, but that is the key of how this termination, row-independent termination, works. Because what happens is, when this side of the stem loop zips up with this side, it's going to be literally yanking from the rest of the RNA transcript. It's forcefully pulling from it to the point that the RNA transcript that is still kind of base pairing with the template DNA gets yanked away, and it gets taken away from RNA polymerase. RNA polymerase is not necessarily binding to the growing RNA strand. It's just going nucleotide by nucleotide, checking if there was proper base pairing between the new incoming nucleotide and the corresponding one in template DNA, but it's not really holding on to any of this, right? So when the stem loop yanks from the rest of the stem, it physically pulls the RNA transcript away from RNA polymerase. And for this to work, for this pulling to be successful in stealing the RNA molecule from RNA polymerase, the binding between that tail of RNA transcript and the corresponding sequence in the template DNA should be weak. So just like the stem reverse complementary sequences were rich in C's and G's, this sequence here is very rich in A's, such that you have base pairing between A's and U's in the growing transcript. So we have this perfect storm between a very aggressive, very violent pulling by the stem loop and a very weak binding between the A's and the U's, which makes it effectively be pulled away from the template DNA. And once the RNA polymerase is left without that growing RNA transcript, like before, it just falls off DNA and finds the next core promoter. Okay? Yes?
So basically what it does is that it's rich in G and C, or guanine and cysteine, and then because of that, it just ends up being pulled away from the rest of the system, and then it brings in what would be the continuing on for the RNA just to stop.
It stops because now the RNA polymerase doesn't have that end of the RNA transcript to add a new nucleotide to. And when RNA polymerase finds itself, here's the thing, RNA polymerase finds itself in unwound DNA without an RNA molecule at the very beginning. But it's fine because it's with sigma, and it's in a space where not having an RNA molecule yet, it's fine. I told you that it can start from scratch. So why wouldn't RNA polymerase start from scratch again at that position? Among other things, because it doesn't have the sigma. And in eukaryotes, it wouldn't be able to do that because it wouldn't have the TF2 family proteins.
But then what happens to the loop after the RNA polymerase goes? Does it unfold itself?
Not necessarily. It can stay like that. So it means that any genes that are in bacteria terminated through this mechanism, remember how I mentioned the other day that even mRNAs will have stretches of secondary structure? So in a way, you said it yourself. So these particular mRNA will most likely look something like this. Right? A long mRNA molecule with a little stem loop at the end.
So it can stay like that, or
It can stay like that for the rest of the life of that mRNA molecule.
So it can also be just like tagged immediately though, to be like broken down and.
It doesn't have to, because in fact, this was just a signal for terminating the transcription of this thing, but this thing now needs to be translated. So no, it won't do anything to this molecule. It's just telling RNA polymerase, oh, you shouldn't bother from here on. So
then will secondary structures always signal like the end?
in the context of this, in the context of row independent termination, yes. Because that the formation of that secondary structure physically steals the RNA molecule from RNA polymerase. Very similar secondary structures, these hairpins or stem loops, might play different roles in a different context. But in this context, the formation, this violent formation, sudden formation of this stem loop that steals the RNA from RNA polymerase effectively functions to terminate transcription. Okay, so those are the two mechanisms that bacteria use to terminate transcription. And it turns out, this thing stayed zoomed in. It turns out that I mentioned a couple times now that eukaryotes do not actively terminate the transcription of their protein coding mRNAs, but they do terminate the transcription of other types of RNA. So RNA polymerase II does not terminate transcription through either of these mechanisms, but it turns out that, there you go, that RNA polymerase I uses an 18-mer, meaning 18 nucleotide signal, that is bound by a termination protein like RAW, and that RNA polymerase III uses an RNA hairpin like the RAW independent mechanism. So both have been conserved functionally, even though the actual proteins may not be the same, but conceptually the method they use is the same. So be aware of that, because it's an easy mistake to make, to say that eukaryotes don't terminate transcription. That's true of protein coding mRNAs. But when it comes to ribosomal RNAs and transfer RNAs, there are mechanisms of active termination that are very much like the ones that you would find in bacteria. Bacteria do it for everything, even their protein coding mRNAs. Yeah? Questions, anyone? No? Cool. So, let's see. 24. All right, well, we will have to move kind of quickly through this. So in bacteria, after chain termination, that's it. They can start translating. And in fact, I mentioned this before, because DNA in bacteria is floating around in the cytoplasm, there's no nucleus. And that RNA that is being transcribed is accessible to all of the ribosomes. The second that the ribosomes see an mRNA, a functional mRNA, and we'll discuss what that means on Tuesday, they can start translating it. So what happens is ribosomes, denoted by these red dots here, would start binding those RNAs and initiate translation, which leads to what we call co-transcriptional translation. Bacteria are all about saving time, resources, doing everything quick, right? So this is a very effective way of, you started transcribing a gene, I'm going to start translating it. And translation happens as the RNA is still being transcribed. With eukaryotes, it's a little different. Eukaryotes need more time to take those RNA transcripts and process them further before they can become mature mRNAs that can be translated in the cytoplasm. So let's introduce that post-transcriptional processing of eukaryotic mRNAs. There is 4 stages of processing. There is what we call 5' capping, 3' polyadenylation, splicing for those genes that do have introns. If you haven't heard that word before, we'll explain in a minute. Not all eukaryotic protein coding genes have introns. There are some intron-less genes, but most do. And then nuclear export. And they're all depicted in this cartoon here, but we'll go through them in more detail. So let's start with 5' capping. 5' capping is a chemical modification of the 5' end of a primary transcript. And it serves 2 purposes. One is protection, and the other one is aiding in the initiation of translation. It kind of tells the ribosomes, here's where the mRNA begins, here's more or less where you should try and translate this thing. Okay? A question for all of you: what could a capless RNA hanging on in the cytoplasm mean for a cell? So before these RNAs make it into the cytoplasm, they will be 5' capped. But what do you think the cell thinks if it finds an RNA in the cytoplasm that doesn't have a 5' cap? What will it immediately gravitate to? Anyone? What could an RNA molecule in the cytoplasm mean for a cell? Trash, I like that. Or worse,
A virus?
a virus, right? A virus. There are many viruses that are made of RNA, and they enter the cell through the cytoplasm first before they can make it into a nucleus. They'll be in the cytoplasm as RNA molecules, but it turns out that those RNA molecules do not have a 5' cap because viruses don't make 5' caps. So there is viral infection, and then there is also this idea that they're trash. What happens is sometimes perfectly fine mRNAs that were at some point 5' cap get damaged, get destroyed, get hurt of sorts, and they may lose the 5' cap. So at that point, the cell treats those RNAs as trash because if they're a viral infection, we better break this thing up. And if it's trash, we might as well recycle it, right? Yes?
Well, you answered my question.
I did? What was your
It
question?
If viruses have the 5' caps.
They don't, because that's machinery that eukaryotes have, and viruses, I mean, the day a virus co-opts that function, we're in trouble. We're in serious trouble. But evolutionarily speaking, for whatever reason, they haven't really figured that out. And if any of you, your career plans are to be an evil scientist out of a basement working on some bioweapon, I just gave you some ideas. All right, so 5' capping, what is it? So it is a chemical modification, bunch of enzymes that will take that 5' phosphate group in the very first nucleotide. We call it base 1 here, but that's the base, and with the ribose and the phosphate, that would be the first nucleotide, right? And that one has an intact triphosphate group attached to it. So there is these enzymes that can recognize that, and they catalyze a bunch of reactions such that now that first nucleotide is bound through a triphosphoester bond, not a phosphodiester, but a triphosphate ester bond, to a very special guanine that is upside down, right? So now this guanine is actually exposing a 3' end. So if in a eukaryotic cell, I should think about making this into an exam question. If in a, I'm giving you the answer, exactly, I'm giving you the answer. If in a eukaryotic cell everything goes according to plan, a good, mature, properly processed mRNA molecule will not have a free 5' end, because the very first nucleotide that is forming that cap is actually exposing a 3' end. So it's ironic, we call it 5' capping. It is the capping of the original 5' end of that molecule, but after proper processing, that mRNA molecule will have 2 3' ends, if you will. I don't want to make that too confusing. So this is just the cap, okay? There is still a 5' 3' polarity to these RNA molecules. It's just that they lose it as part of the capping process. And that's it. It's an upside down guanine that gets attached through this weird triphospho triester bond to the 5' end of the molecule, and that marks that molecule as being one of the cell's own. It's basically telling the rest of the cell, don't bother with this one, it's not trash, it's good, it's in good shape, and it's one of our own. Okay? Then we have 3' polyadenylation. We went through this a couple times, but now let's take a look in a bit more detail. So the 3' poly-A serves 3 purposes. Protection. It's important for nuclear export. Nucleus will not export an RNA until it sees a properly formed poly-A. It doesn't bother with RNA molecules that don't have a poly-A. And it is important for translation start. And that's counterintuitive. My question to you is, why would the poly-A be needed to start translation if it is located after the sequence to be translated? Why would the cell care to see a poly-A in an mRNA molecule to start translating it? Why do you think? Yeah.
Does translation go from 3' to 5'?
Oh, great idea, great hypothesis. If translation goes from 3' to 5', then it makes sense. You need to see a poly A that will tell you that that is the beginning of the but no, it goes from 5' to 3'. In fact, that's why the 5' cap is important for translation. That's what tells the ribosome this is the beginning of the mRNA. So why would ribosomes care about the end of an mRNA? Yeah?
Is it viruses again?
Partly, but that would explain the protection side of things. So the poly A protects mRNAs because it's another way of telling the cell, I'm not a virus, I'm not a virus. Right? Goes back to the idea of the trash. The idea is, if an mRNA lost for some reason its poly A, no matter how legit it was at the beginning, if it doesn't have a poly A, the ribosome won't bother with that molecule because it won't start translating something that it doesn't even know if it's complete. What if this mRNA molecule lost the poly A to some sort of damage that took out half of the protein that needed to be translated? Right? If I give you a book with instructions and you can tell that half the pages are ripped, you're like, well, I won't even start with this process. Why would I follow instructions that I know are incomplete? Same here. So evolutionarily, there is this mechanism that said to ribosomes, translate messenger RNAs that have a properly formed 5' cap and a properly formed poly A. Anything else, don't even bother. And this is enzymatically how it happens. So when the RNA gets transcribed, the poly A signal in the polymorylation signal sequence in DNA doesn't really do much. There's nothing binding to it in DNA. But like that rat sequence, the second that it shows up in an RNA molecule, some proteins notice. And in this case, it is these proteins, CPSF and PAB. You don't have to remember those details, but it's two enzymes. One of them cuts the RNA, and the other one, the polyadenylation protein, that's the one that starts adding A's that are not based on a template like RNA polymerase does. It just stitches A's together one after the other. And it will do it for as long as it can. Some poly A's are in the tens. In length, some of them are a couple hundred A's. So these are really long poly A tails.